Clarity Clinical Solutions.
Imagine spending years collecting data from thousands of patients across dozens of clinical sites, and then one day, you declare it final. That day is the database lock. It is the formal act of freezing a clinical trial database so that no further edits, corrections, or additions are permitted. Once the lock is in place, the data is handed to the biostatisticians for unblinding and analysis. It is a pivotal milestone that separates data collection from data interpretation, and getting it right is essential to the integrity of the entire trial. A database lock is the process of finalizing the clinical trial database after all data has been entered, cleaned, and verified. It means the database is declared complete and accurate, with all queries resolved, all discrepancies addressed, and all data points confirmed. From this point forward, the database becomes a read-only archive. No changes are permitted without exceptional justification and formal documentation. The locked database serves as the single source of truth for all statistical analyses, regulatory submissions, and study reports. Database lock is critical because it prevents data manipulation after results are known. If analysts could edit data after seeing unblind results, the trial's scientific validity would be compromised. The lock creates an immutable audit trail that regulators trust. Without it, there would be no way to prove that the data wasn't cherry-picked or adjusted to favor a particular outcome. For regulatory agencies like the FDA and EMA, a properly documented database lock is a non-negotiable requirement before any submission can be accepted for review. Before the database can be locked, a series of rigorous activities must be completed. Data managers perform final data cleaning, checking for missing values, outliers, and logic errors. All case report forms are reviewed and signed off by investigators. External data, such as lab results, ECG readings, and imaging data, must be reconciled and integrated. Serious adverse events are verified against the safety database. Every data point is traced from source document to the trial database to ensure complete accuracy. Throughout a clinical trial, data queries are generated when entries are unclear, inconsistent, or missing. Each query must be resolved before database lock. Data managers issue queries to clinical sites, and site coordinators investigate the source documents to provide corrections. The goal is to achieve zero open queries at the time of lock. A query resolution report is generated, documenting every question raised and how it was resolved. This audit trail is crucial for regulatory inspection and ensures the dataset is as clean as possible. Data quality checks are automated and manual processes that verify the completeness, consistency, and accuracy of the clinical data. Logic checks ensure that dates are chronological, for example, treatment end date cannot precede treatment start date. Range checks flag values that fall outside normal physiological limits. Cross-form consistency checks verify that a patient's data matches across all case report forms, while missing data patterns are carefully analyzed. These checks are documented in a formal data quality report that becomes part of the trial's essential documents for regulatory inspection. The database lock meeting is a formal gathering of key study stakeholders to authorize the lock. Attendees typically include the data manager, biostatistician, clinical lead, medical monitor, and a representative from clinical operations. They review the data quality report, query resolution status, and any outstanding issues. Each attendee confirms their readiness for lock. A formal sign-off is documented in the meeting minutes. This meeting ensures that everyone with responsibility for the data agrees it is complete and ready for analysis. Database lock is a team effort. The data manager leads the cleaning and validation process and prepares the lock documentation. The biostatistician confirms that the data structure supports the planned analyses. The clinical data manager certifies that all data has been entered and queries resolved. The medical monitor verifies medical coding and safety data. The clinical operations lead ensures site-level activities are complete. Each role has defined sign-off responsibilities documented in the study's data management plan before the trial begins. A comprehensive checklist guides the database lock process. Key items include all expected case report forms received and entered, all data queries resolved, external data reconciled, serious adverse events reconciled with safety database, medical coding complete, audit trail reviewed, data quality report generated, and all study team members have provided sign-off. Each item is checked and dated. The completed checklist is signed by the data manager and filed in the trial master file. Regulators frequently request this checklist during inspection. Once the database is locked, the focus shifts immediately to analysis. The biostatistician receives the locked dataset, performs final validation checks, and then executes the pre-specified statistical analysis plan. The database is archived in its locked state, preserving an immutable record. Unblinding occurs according to the study's blinding and randomization plan, and clinical study report writing begins promptly. The locked database and all associated documentation are prepared for potential regulatory inspection. Any post-lock changes require a formal amendment process with documented justification and senior-level approval. Database lock directly enables the statistical analysis that determines the trial's outcomes. The locked dataset is imported into statistical software like SAS or R. The pre-specified statistical analysis plan is executed, producing tables, listings, and figures. The results are reviewed by the unblind study team. Because the data is frozen, regulators can be confident that the analysis was performed on a fixed, immutable dataset. Any discrepancies between the locked data and source documents would be a serious finding during a regulatory audit. Regulatory agencies expect a clearly documented database lock procedure. ICH E6 Good Clinical Practice Guidelines require that all data processing activities are documented and that the database is locked before analysis. FDA's 21 CFR Part 11 governs electronic records and signatures, requiring audit trails that show who made changes and when. During an inspection, regulators will review the lock documentation, query resolution process, and any post-lock changes. A poorly documented lock is a common inspection finding and can delay regulatory approval. Several pitfalls can delay or complicate database lock. Incomplete data entry from slow sites is a leading cause of delay, often requiring intensive follow-up by clinical monitors. Poorly designed case report forms generate excessive queries that are time-consuming to resolve and frustrate site staff. Late-breaking serious adverse events may require last-minute updates that ripple through the reconciliation process. Reconciliation mismatches between the clinical database and external data sources like central labs can hold up the entire process. Finally, inadequate documentation of the lock process itself can lead to significant regulatory findings during inspection. Database lock is the critical transition from data collection to data analysis in a clinical trial. It ensures data integrity by freezing the database and preventing post-hoc manipulation. The process involves thorough data cleaning, query resolution, quality checks, and formal sign-off from all stakeholders. A well-documented lock procedure satisfies regulatory requirements and provides confidence in the trial's results. As clinical trials grow in complexity, robust data management practices, including proper database lock, become even more essential to delivering reliable evidence to regulators and patients.